From ASM Microbe 2023, this is TWiM, This Week in Microbiology, episode 290, recorded on June 18th, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today in Houston, Texas, Michael Schmidt.
Hello, everyone.
Michael, what's the weather like outside?
As I just told during the warm-up to our audience, you wouldn't know it in the convention center, but there's a heat advisory out in Houston, and it's going to be that universal temperature, 37 degrees or 98.6 today.
All right, thank you for the weather. Also joining me, her first live TWiM, our latest addition to the team, Petra Levin. Welcome.
It's great to be here in Houston at ASM. And it is cold in this conference hall, though.
This is very cold here. They keep it cold in Texas. We have a very special guest for you today. She is a professor emerita at the University of Texas, also an adjunct at McGovern Medical School. Mimi Goldschmidt, welcome to TWiM.
Thank you.
Have you been to one of these meetings in a while? Or is this the first time?
Not in a little while, for
Been a
about
while.
5 years.
5 years. That's not
4
so
or 5 years.
Okay, well, we're happy to have you. We're going to talk a little bit about your career. Let's go way back.
Let me go way back because I grew up a little differently. I was born in 1926. I was 97 a few days ago. 97. I have had a stroke and I'm not mobile as much as I should be, but my mind seems to be fine. I would like to tell you a little bit about the history before I came. World War II was 1939 to 1945. Maybe a lot of you weren't born then, so you don't know much about World War II. Have any of you heard of Rosie the Riveter? Oh, good. I see a few hands going up. And saddle shoes. Yeah, I wore saddle shoes through graduate school. There was rationing when the war was on. There were things that weren't rationed that you couldn't get. You couldn't get tires. You couldn't get automobiles. You couldn't get refrigerators or washing machines. Those things were all converted to war effort. But they had rationing, and you had a little book full of coupons. And you also had little coins, little red coins that you could use. Now, I'll tell you what was rationed. Sugar, gasoline, trees, meat, coffee, butter, shoes. Shoes were rationed. That means that you couldn't go out and buy yourself a couple of shoes. There was no silk. It was all going to parachutes in the war effort. So you really couldn't find decent stockings to wear. There are no more silk stockings, which were wonderful when we first used stockings. But now women just kept in their bobby socks and their saddle shoes, all through graduate school. And that is the way that I grew up. Part of the war was on when I was partly in college. I had to take my ration book to college and so forth. And then during my first 2 years in graduate school, I had to take my ration book with me. So that's about how I grew up in a war effort. I was a Girl Scout, and we knitted bundles for Britain and so forth at our Girl Scout meeting. And we went out and collected people who smoke cigarettes. The inside of the cigarettes is our tinfoil, and we made big bags of tinfoil. my mother would save grease and she'd have to turn it into the butcher and things like this. And soap was not rationed, but there wasn't very much soap around, and hair shampoo, fancy stuff, none of that was available.
So Mimi, where did you go to undergraduate, and then what made you select your graduate school?
I went to Western Reserve University, and I had a scholarship to college. So I avoided taking microbiology because I never think I would spell all those names. And the dental school has some that are really long. But I fell in love with microbiology as a graduate student. Now, I knew when I was 10 years old and I had been to Girl Scout camp, and the person who was the nature counselor had put up all sorts of questions so that I got very much interested in biology. And that's how I knew I was going to be a biologist when I was 10 years old. That was it, biology. And it wasn't until I was a junior in college that I was fixated on microbiology. I figured, oh, what a wonderful field and what fun it is to do and so forth.
So I have a follow-up, which is, how did you decide to go to graduate school? Because it wasn't...
Let me tell you about graduate school.
Do we want to hear it?
We do.
My teacher said I should go to graduate school, and they named some of the best graduate schools for microbiology: Wisconsin, Purdue, a University of Pennsylvania, and so forth. So I went home, and after my mother fed my father a good meal, I looked at him and I said, I should go to graduate school. He says, no way are you going to graduate school. He says, nobody is going to marry a woman with that much education. And my mother said, that's the kind of man she should marry. And then my uncle, who was there, said to my father, well, she may be a spinster, but she will be able to support herself. And on that premise that I could support myself, my father said, yes, you could go to graduate school. University of Wisconsin, too far away from home, my mother says. No, Wisconsin. The next school was Purdue. I said, that's just beyond Indiana. And she said, okay. So I went to Purdue for my graduate work. And I worked under Henry Koffler for my master's degree. A woman had to be serious about going to college and going to graduate school. So women who went to graduate school had to get a master's degree first before they could work on a doctorate. So I worked on Henry Koffler. I worked on the production of penicillin by penicillin chrysogenum. And I looked at the shake flask. Penicillium is a fungus, and it grows as a mycelia, long snakes. But I had had enough chemistry in college that it looked to me like it was foaming, and I added things that cut down on foam, and I added oleic acid, and it grew as little tiny yellow pellets about the size of my fingernail, and the amount rose up and quadrupled the speed to start with, quadrupled the amount of penicillin there. Now, the person who I was working for, Henry Koffler, had gotten his degree at Wisconsin, and he was 2 years at Purdue. He wasn't tenured or anything. And here I was finding a wonderful way to produce penicillin. And he took it as if he had decided it and had put me on it. He never gave me any credit at all. And publish papers with just his name on it. And this happens to a lot of women when they are in graduate school. Their professor takes complete credit for anything they may have come up with. And I never really had credit. And we theoretically published a paper when I finished my master's degree, but my master's thesis will state that there is a reference for Koffler first and Goldschmidt second in the American Journal of Botany, but it was never published for some reason. So that was my master's degree. I started to work on my PhD. I'll tell you something. If you read the book that Rita Colwell wrote, she went through Purdue and had the same problem with Henry as I did. The business of being transferred and transferred. In the meantime, my husband was working right away on his PhD, Eugene Goldschmidt. And so I switched major professors, which was pretty unknown at that time.
So you didn't end up a spinster?
Yes.
You did not end up a spinster?
No, I didn't end up a spinster. I ended up married, much to the unhappiness of university. This is another story. The university was very, very upset that I got married. They were even more upset when I had children. I dared to have children. I have two wonderful children. My son has a PhD in neuroanatomy and brain function, and my daughter is a lawyer in the tax area. And when Purdue finally decided to make me one of their outstanding alumni, and I took my two children back with me. They were adults by then. And I said, this is Dr. Goldschmidt, my son, and this is my sweet daughter, who's a good lawyer. And then they gave me the degree, and they asked me to help. Purdue asked me finally to help them decide on how to keep young professional women to stay at Purdue and things to do. And let me tell you, they completely changed from when I was a student there. And we planned to let them extend the time for them to be considered for tenure. And I said, you've really got to get on this campus. It's big enough and there are enough departments and a lot of engineers to have women bring their children so that their children can stay at a place right here on campus. They won't have to drive around or at 4 o'clock leave to go and pick up their children. So Purdue did that finally, connected with a day school, babysitting for children. So that was something that I accomplished that I was very happy to. And I told them that women should be considered serious when they come to graduate school, but they are also a woman and they're also a scientist. I said, and a man is a man and he's also a scientist, and nobody really looks at this difference. I've had people tell me I can't make as much money because my husband is working and getting a salary, so you're not being paid as much as people that you should be paid. And I said to them, what does my worth as a scientist have anything to do with whether I'm married or single? And this guy just pushed his grant back at me and said, that's it, take it with $2,000 less money or not at all.
When did these kinds of attitudes change in your whole career? Have you seen that?
I think it changed when the ASM, I think first of all the war changed, and women who were in their home were able to move out. I mean, women were secretaries or they were nurses. They couldn't become doctors. Finally, when the war came on, women moved out into industry. Rosie the Riveter is a good example. She will show her muscles, and women weren't supposed to have muscles like that, and so forth. And women were supposed to be lovingly and protected in their own little houses and raise their children. Well, raising children is important, but you can do it and do science too. And
well, that's quite evident that you raised a neuroanatomist and a tax attorney.
Yes.
Two very challenging things for children to aspire to. So
and I had to psych out my kids all the time.
So,
that's the one thing, having a brain. You know how to treat them in a way that will make them happy with what they're doing, but not doing things. My son came home one time and said, "I'm going to be a Black Panther." And I looked at him and I said, "Well, what does a Black Panther say? Let's read it and see what they say." And he got information about the Black Panthers, and I saw that he threw them in the garbage, and he said, "I don't agree with enough of their principles to become a Black Panther." And he decided that himself. So
let's transition to some of the science you have done over your career.
Yeah.
Because the audience may not know, but Mimi was at the front door of the development of biosensors. So why don't you tell a little bit to this audience about how you got into the field of biosensors
let me talk about
any of your science.
My 2 years at NASA. I was working at Baylor part-time. When my kids were in school, I was in the lab. I worked with Bob Williams, who was later a president of the ASM. And they were trying to find a person who would go to NASA for 2 years. And I said, "I can do that. My husband is tenured. I can play for 2 years." So I went to NASA as the director of the protocol to plan the Lunar Receiving Laboratory and to look at what the first moon rocks that came back. I knew Buzz Aldrin's and Neil Armstrong had been gotten his degree at Purdue, so he was a little familiar to me. The business of NASA was like every other government institution. They didn't want to report that they hadn't spent all the money the government gave them because they were afraid they wouldn't get a bunch more. And I had to report that to the university and so forth. They were not too happy to hear me report that, but that is the way it happened. Now, I noticed something very interesting at doing the research and setting up the tests. We used notobiotic animals, and we used pieces of plants, and we used bacteria and a whole other fungi and other anaerobes and aerobes to see whether they had any effect on bacteria. The material is like soil and rocks, and it really affected plant growth and enhanced it, but it didn't do anything as far as killing animals or killing people. And we also planned to put the astronauts in for a 2-week incubation period in case they brought back anything from the moon, which they didn't. I noticed that all of this was Petri plates and no rapid methods, and so I figured this is an area for me to go into, is biosensors and rapid tests and so forth. Now, I must tell you, I came to the ASM and I was invited to speak at a clinical symposium. And I was very honored. And this was early on, about 2 years after I left NASA. And I was introduced like this. This is Dr. Mimi Goldschmidt. She's going to talk on rapid methods related to physical aspects. And then the director said, she's not only our token woman, but she really knows what she's going to say. And that was my introduction. And I really knew what I was going to say. And I got back to my seat. The man sitting beside me was Dan Fung, who's a clinical, well, he's more of a food microbiologist. And he was chuckling and he said, yes, you really know what you're going to say. He says, you're going to have to come out and lecture at Kansas State to the food microbiologists. So for 30 years in the summer, for a week and a half, I joined Dan at Kansas State University in a rapid detection of microorganisms that are related to food, so that I know how to go from farm to fork, and it enhanced my eating as well.
So can I ask, how did your detection method rely on chemical tests, or how did you detect the microbes? Like, what is your,
if I have my choice, I think an immunologic test is a very, very fast way to detect bacteria, especially those in a mixture. You don't have to sort them out on a Petri plate first and get pure colonies, although pure colonies are much help at the end. So I think immunology, I did work on amino acids and so on and so forth, and I looked at all of these methods from physical on, and if you write me, I'll give you a reference to 2 or 3 books that I have added chapters to on this area. But I really think immunology methods are good. And the last book that I wrote, I included even the small neutral particles are really good in biosensors and rapid detection methods, and which was important to me and important to people in medicine.
So you mentioned Neil Armstrong and Buzz Aldrin. Did you get to meet them?
Yeah, I have met them. Now, Buzz Aldrin was an interesting man, and we had to get a microbiologist who had been a wrestler to teach him how to use aseptic technique, because he had a sterile shuttle and sterile bags, and he had to scoop up some of the first moon rocks to return, and he had no idea of sterility or not breathing in the bag that he is putting things in, and not to take it out of footsteps that are there on the moon already. And you people all know about how to protect where you sample, as far as the kind of land or what you sample. But this was lunar rock that he was sampling.
Because we didn't send the astronauts to the moon sterile. I mean, they were real people.
They were real, and they were reeking by the time they came back, because they didn't take baths, the first group of people. They off-gassed, those suits off-gas. So I'm sure they have contaminated where their footprints were, and the moon is probably contaminated with bacteria from the astronauts.
So the Sea of Tranquility actually now has microbes.
It has microbes.
From us.
And if there were any anaerobes, they have probably survived.
Probably chemolithotrophs. Probably chemolithotrophs, I would bet.
Right.
But it's probably the radiation that has done them in.
The radiation has done them in to some extent. I will tell you about another study that I did before. I worked at Fort Detrick for a couple companies there. One company was interested in what happens, can they use bacteria in a bomb someplace? But they would blow up bacteria and they would see that the light from the bomb was enough to kill the bacteria, so they wanted to see what the shock wave did. So if you can imagine me pregnant with my second child, about 6 months, I was in a hard hat and there was sandbags and a big plunger, and I blew up pillows of bacteria that were attached to a little parasite, a little parachute, and we took them back to the lab and plated them out and then looked at them under the electric microscope, and they were riddled, bubbled. Cavitation is what occurs with shock waves. And frankly, it's the same thing that occurs with IEDs of our soldiers that go to Afghanistan and get hit with an IED. And I have made suggestions about this, and I knew that there were covers that people have used under football helmets that protect them from having concussions. So I suggested that they try a kind of cover that will prevent a lot of this
Sonic
agitation
energy
and
from
the
coming in.
cavitation that occurs. But that was interesting to be pregnant, and I've had a lot of fun.
But you know, did you think that plunger actually then influenced your daughter to becoming a tax attorney?
Yeah, it could be. Well, you know,
Could be.
my family had mathematicians. My mother, who was stopped by her father from doing anything except to be a secretary. She had a very, very good mind, and she was a really good mathematician. And two of my uncles were good mathematicians. So my daughter was a really good mathematician. And she graduated summa cum laude and then went to law school. And law school has invited her to be an adjunct professorship at the law school to teach tax, which was very nice. But she likes the job that she has, and today she would be here, but she's flying out to California for her company. She's a partner in a big audit firm, all that has buildings, and she goes and teaches tax all around the country.
Well, she can watch you on the video and after. So,
Oh, she can?
yeah, you bet she can.
You have some more questions.
So do
Vincent.
you follow the space program anymore?
Yes, I do. In fact, last year I added to a publication. About 30 people added comments to. So last year I had a publication. This year I'll have a publication for some work that I did, and a graduate student did something last summer, and we're publishing another paper. So I'm still involved in publication and working. I assist and help somebody else in their lab. I don't have a lab. That's very frustrating because I have very good ideas on projects to do, and I don't have a lab to get into.
You know, we're going back to the moon, right?
What?
We're going back to the moon.
We're going back to the moon, and they're going to try to live there for a while.
So if you were younger, would you go to the moon if they asked you?
Would I go to the moon if they asked me? Depends on the age, yes. But the problem with going to the moon is you got to dress up, and it's anaerobic. So you have to wear all sorts of gear. And I imagine if they build a building that they can put air in, maybe 21% air, oxygen in a building that people could do research up there because the gravity is different and so on and so forth. So I suppose people will be working on the moon eventually, and the ASM will be involved because there's water on the moon, there's water underneath the moon, and when you see water, you'll see bacteria. So I think that they're going to find interesting organisms that are probably can be anaerobic as well as aerobic and protect themselves from the sunlight.
Do you find Mars or the moon more exciting in potential?
Do I what?
Is Mars or the moon more exciting in the potential for interesting microbes?
Oh, I think there's potential for interesting microbes, and I really don't think you're going to find much difference in the microbes that we have on Earth or the microbes that are underwater and so forth. That's an interesting area I didn't get involved in, is the bacteria in the deep sea cliffs that were living off of the smokers that are using the energy, the chemical energy to grow and to grow there. And I think that is interesting research for those kind of organisms.
The smokers are the ones that are emitting highly enriched sulfur compounds.
Right.
Along with a few metals. And those
Right.
of you interested, Norm Pace did a bunch of work on that in the early days when 16S was just getting started.
Right. You're going to have to remember that the reason we're in this room and breathing is because of the bacteria. You're 2.6 billion years ago, all of a sudden bacteria and sunlight were able to combine, and the production was oxygen and CO2, but oxygen became a weapon for bacteria. Bacteria really were the first ones that started warfare. They produced oxygen, which was a weapon against the anaerobes. And they also produced enough oxygen over billions of years that you and I can breathe, sit here and breathe oxygen due to bacteria. And I think I don't understand why the whole world isn't filled with microorganisms and with people. The microorganisms are there. The people are here working on them, and that's so much fun, and it's so important to work. And you make the world better by the work that you're doing. And I think you have an ability with yourself as your profession to contribute to the world and to contribute as a woman. I have contributed all the time to helping other women go through what I went through, and I hopefully they're doing better. And I was honored by the ASM to be a member of the Alice Evans Committee, and I see at least one in the audience, Lorraine Finley there, who was elected also. And I worked with the women's group. In fact, I was one of the first women at a meeting that decided that they were going to have a women's group, which they need. And I've been very happy working with the ASM. I was involved in administration as well as a member of what we used to be called the CPC, the Council Policy Committee, and I have chaired committees. And I think, the first time I went to a microbiology meeting was at the University of Minnesota, and it was a hotel in Minneapolis. And I walked into the door, and I only knew the people from Purdue that were there. And I saw all these people talking and laughing to themselves, and I thought to myself, will I ever get to know people? And one way to do this is to volunteer. And know the people. And another very, very important thing is to be active in your branch and take your branch and help it out. And I see members of my branch here that are just wonderful. And Texas has a good branch. And we contribute and we help students and so forth. So it's important for you to help your branches, to help the ASM, to volunteer. The minute you volunteer, you make wonderful friends that will last you for life. I know I have friends here that I like, Lorraine and Heidi and Bob, and it's one thing to get involved. I know I'm 97 and I'm home most of the time, but I'm on the computer. I still sit on the board for the branch, and I sit on the board for the Rice University Texas Medical Center chapter of Sigma Xi. That's another interesting area, is Sigma Xi, because it'll spread you out. And I have worked with problems with engineers and so forth that I never would have met or gotten around it, so that my work on biosensors has extended to doing engineering too. And the more, and one thing I'll say about volunteering, if you think the ASM needs something that isn't there and you can provide it, talk about it, speak up, and get yourself involved, even if you originally start the project. So,
Can I ask you one last question?
yes.
If you had not been a microbiologist, what would you have been?
I was interested, before I was involved in microbiology, I was interested in what was happening when things were growing, like babies and so forth. Embryology interested me at the time, because I felt that embryology and having a baby was such a miracle, that the people had so many babies that were okay, and that was interesting to me. And I think I could have been an embryologist. But microbiology is so satisfying, and I've had such a wonderful life. I didn't retire until I was 85 because I was having such fun. And I never told the chair of my department I was having fun, because my bonus, my payment was really a bonus. And my husband and I were on our way to talk at a meeting in Czechoslovakia. I have traveled as a microbiologist. That's one thing I'll say. Have slides, will wander. And I've talked in South America, and I've talked in Mexico, I've talked in England, and I've talked in Scandinavia, and I have had a lot of fun meeting people and taking vacations along with it.
And Mimi, wasn't your husband a virologist?
My husband was a virologist, yes.
Vincent's are like that.
I do. I like it very much.
My husband was a virologist, and a wonderful teacher, Mimi Goldschmidt. He died at 56 in 1980, and it was terrible. It was just terrible. I still miss him, and if I pick up a camera, I hear his voice saying, "Now focus, don't focus halfway on a picture," and so forth. I hear him talking to me when I pull out a camera. That was his hobby.
So, does anyone have a question you'd like to ask Mimi? Because we do
we
have
got lots of questions.
All right. Ray will get you a microphone.
Hi, Dr. Mi. That's wonderful talk, and that I give a lot of thought to women's research, also everyone's research. I have a question about, so when you doing research, you follow your heart and your interest, or you follow where the fund go?
What's the question?
Do you follow your heart when you're doing? Do you follow your heart when you're doing research, or do you follow the funds?
I follow my heart, and I hope to find someplace funds. But I do. You have to do preliminary research before you can apply for grants or anything. So I have had some weird grants.
Okay, another one.
Thank you so much for your talk today. It has been an honor and a celebration of life to hear you speak. It seems to me that you went through a lot of adversity at the beginning of your career, and I was wondering if you had a word of advice, particularly for scientists that are parts of minority groups, to persist in science in spite of adversity.
So you've gone through a lot of adversity in your career. Do you have any
Yes.
advice for people who might face the same?
I'm a survivor. Let me put it that way. I'm a survivor. I've had a lot of prejudice. At one time, I ran the clinical microbiology laboratory at MD Anderson Hospital. And I noticed that the good old doctors were sending specimens, but they really didn't care whether they were resistant or whether they were sensitive. They used the same antibiotics and the same antimicrobials, regardless of what they had sent. So I wrote them a letter and told them that I hoped that the lab results were helping them with their treatment of patients.
So you were a pioneer of antimicrobial stewardship.
At the hospital, I brought computers into the lab for the first time, into microbiology, to set up some way to keep track of what was there and so forth. I started the computers. But the doctors were very, very upset, and they told the head of the department, and I quote what he told me, "We don't want any woman PhD teaching us how to practice medicine, and you're fired." So I was fired to run the lab, but I still there and continued the research on rapid methods that I was hired to do, actually. But I have had that in more than one situation where a woman was, it's very hard for a woman to be taken seriously. At least it's better now. And I think the ASM is very helpful to women here. I see a lot of women. The last time I spoke, I was telling Michael that we used the fancy word biome. And Lorraine and I were on the same program. I was talking about the oral biome. This is the organisms in the mouth and so forth. People were talking about the skin biome, and the word biome is so fancy. It's become a fad word. I don't know whether they're still using it or not.
They still are. We have one more question, Mimi.
Going off of that, thank you. Your talk has been very inspiring. So do you have any advice for like young female scientists starting their careers?
Starting your career. I think you don't have to show initially the complete interest in microbiology. I think you need a lot of chemistry, a lot of biochemistry, a lot of science, and then you maybe should make up your mind of what kind of science you're interested in. You might want to even just be a biochemist, or you can be a biochemical microbiologist. Now, when I was a junior in college, I decided that I was going to be a microbiologist and that I needed biochemistry. The only biochemistry that was taught was at the medical school. So the only women that they usually allowed to take biochemistry were nutritionists or people working with food. So I hiked myself over to the medical school and made an appointment to talk to the head of the head of biochemistry, and he asked me, would I be a nutritionist? No. And I said, no, I'm going to be a microbiologist, and it's not too soon to start biochemistry. He said, I'm going to make an exception in your case. And he let me take biochemistry. Again, I was the only woman there, and I was a non-person until about almost at the close, when we had a big experiment where you filled your jug with urine after eating a certain diet. I got back into the laboratory with my little jug and set it on the table. And the future doctors of America are whispering in the corner. And finally, one came to me and said, how did you get your urine in the jug? And I said, I used a great big funnel in a loud voice.
All right.
All right, I think that's
So, but from then on, they talked to me.
That's a great way to end this episode of TWiM number 290. Show notes are at microbe.tv/TWiM. You can send questions or comments to twim@microbe.tv. If you'd like to support our work, microbe.tv/contribute. Our guest today from the University of Texas, Dr. Mimi Goldschmidt. Thank you so much.
My pleasure. I'm honored. It's an honor to be here and talk. And thank you for inviting me.
You're welcome.
I'm really honored.
Michael Schmitz at the Medical University of South Carolina.
Thank you, everyone.
Petra Levins from Washington University. Thank you, Petra.
Oh,
Thank you.
thank you.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology and the American Society for Virology for their support of TWiM, and Ronald Jenkins for the music. This episode was recorded and edited by Ray Ortega, who's standing right there. Thank you, Ray.
So you now can all see what Ray looks like.
Thanks for listening to This Week in Microbiology. We'll see you next time.
Good. Thank you.
Thank you, everyone.